Hi. So, in previous session we have learnt about the antibody structure and function, and those structure was basically elucidated with the help of simple biochemical methods. Biochemical methods includes the electrophoresis and the purification by chromatography, and then generation of antibody against specific fragments. Okay. So, today in this session we will look at various kind of antibodies and how what are the different types, as well as how they are structurally different, and when they are structurally different, then their function is also different. Okay. So, when this antibody structure was elucidated, then there at that time the protein sequencing was also developed. Okay, if you remember, there is a, if you studied the biochemistry, you can understand there is a protein sequencing method was also there at that time. So, when amino acid of light chain were sequenced, so since the technology is there at that time, so they found out some interesting thing. First and foremost important thing is that the first N-terminal 110 amino acid, they are having lot of variation. Okay, and this variation is as now you know that this region is needed for the binding of needed for the binding with the antigen. Okay, and other part remain constant. Okay, so this other part is remain constant, means the sequence is not varying. Okay, not varying too much, but they found out that there are two kinds of this sequences there. Okay, and these two kinds of sequences, which is predominantly present in almost all antibodies which are which were sequenced, they are either kappa type or lambda type. So, they gave a name, simple, kappa and lambda is the name. There are two kinds of sequence, and these two kinds of sequence, they call it as a kappa or lambda. So, whatever they have sequenced, the constant region is either kappa type or lambda type. Okay. So, with this this was again emerged that this light chain is having a highly variable region, which is consist of approximately 110 amino acid, and rest of about 100 amino acid, they are constant. Okay, approximately 100 amino acid. If you see the structure, the light chain is consist of 214 amino acid. So, first 110 is variable, and rest of 100, approximately 100 amino acid, they are either kappa type or lambda type. There are two categories of sequences. Okay, and when these work are sequenced, then they found out that in human the 60% of the constant region in light chain is kappa type, and 40% falls in lambda type. Okay, and in mouse it is about 95% is kappa type, and only 5% is lambda type. Okay. So, this was a quite important information about the antibody, because if you see that this antibody has a variable region and constant region. So, later on people wanted to address this thing based on the genetics. Okay. So, I will talk all those genetic component later on. So, the people also sequenced the heavy chain. Okay, which is consist of about 450 amino acid, and they found out that again the N terminal show a hypervariable variation in the amino acid, which is approximately 110 amino acid, and rest of the sequence they found out that these rest of constant region are 5 different types. Okay, and this 5 different types of heavy chain we call it as a isotype or subtype of antibody. Okay, and these are basically the chain which is present in these heavy chains. They are mu, gamma, alpha, delta, and epsilon. Okay, so they are whatever they have sequence, they found out that most of this constant region fall either of any of these classes. Okay, and that was the basis for the different kinds of antibody. Okay, some sub isotype have also discovered. For example, you will see in this session that IgG, which is having a gamma heavy chain, they have little differences, and these little differences is result to the formation of different subclasses. For example, IgG1, IgG2, something like that. Okay, so we will discuss all those things in and this is not only in human, this is also present in another species, particularly mouse. Okay, each antibody's body has two identical heavy chain and two identical light chain. That I have explained you in previous session, that these antibodies are basically dimer of heterodimer. Okay, so this dimer of heterodimer is basically makes a complete antibody. Okay, so now I will show you what kind of antibody could be there. There is some kind of formula, molecular formula for each antibody. Here you can see that in extreme left column there is a classes, that is IgG, IgM, IgA, IgE and IgD. Okay, they have a different heavy chain like gamma, okay, gamma, mu, alpha, epsilon and delta, and there are in some cases there are some subclasses also. For example, in case of IgG there are 4 subclasses like gamma 1, gamma 2, gamma 3, gamma 4. In case of IgA also there are alpha 1, alpha 2 subclasses. Okay, and these heavy chain can either interact with kappa chain or it can interact with lambda chain. Okay, so here you can see that in all classes of antibody, okay, it is either kappa or lambda. So then the molecular formula could be here you can see that gamma 2 kappa means there are 2 the heavy chain is 2 gamma heavy chain and 2 kappa chain. It is one possibility. Another possibility is 2 gamma chain along with 2 lambda chain. So this is kind of a molecular formula. Okay, and here you can also write in case of IgG, it could be gamma 1 2. Okay, gamma 1 2 means there are the subtype will be gamma 1. Okay, and it can combine with either kappa or it can combine lambda. Okay, so then it will be a gamma 1 2 kappa 2 or gamma 1 2 lambda 2. Okay, so like that I am just trying to make you understand that this is not so complicated. If you see carefully and logically, then you can understand quickly. Okay, here you can see that IgM, they are basically present either in monomeric form or in pentameric form. Okay, here you can see that in bracket there is a N, and N could be 1 or 5. Okay. In case of IgA, this can make a monomer, or this can make a dimer, trimer or tetramer. Okay. And rest other antibodies, they do not make a multimeric form. Okay. So, these are the information which is very important for understanding these antibody molecule. Okay. So, if you look at this formula, you can understand that there are how many subtypes, whether it is present in monomeric form or multimeric form, all those things. Okay. Now, I will talk about individual immunoglobulin. I will put it in one group. Here you can see that there is a IgG and Ig delta and Ig. So, IgG is basically encoded by gamma chain. Okay. So, IgG, IgD and IgA, which is encoded by gamma, delta and alpha respectively. Okay. So, all these three classes of antibody, they have some unique region in constant region. Okay. In heavy chain, they have a hinge region. Okay. It is a hinge region, and this hinge region is present in heavy chain. Okay. And so, hinge region is nothing, it is basically having an extended amino acid. There are more amino acid, and they have some characteristic amino acid which I will show you. So, this hinge region is basically rich in proline and cysteine. Okay. Since it is rich in proline and cysteine, so this has a more flexibility. Okay. And this can also make a more disulfide linkages. Okay. So, you remember that in case of IgG, Ig delta and Ig alpha, this hinge region is present, and this hinge region is nothing, it is a more amino acid which is predominantly consist of proline and cysteine, and this gives the flexibility to the molecule. Okay. In these classes of antibody, or in these isotypes of antibody, there is a CH2 and CH3 region, and this CH2 and CH3 region is quite heavily glycosylated. There is a lot of oligosaccharide side chains, and it is considered that these side chain basically play an important role in keeping these antibody in soluble form. Okay. So, these these sugars are kind of hydrophilic, and this keeps the molecule in soluble form. Another is IgM and IgE, which is encoded by the heavy chain is encoded by mu and epsilon chain. Okay. They are present in secreted immunoglobulin, and it is generally present in membrane bound immunoglobulin. Okay. So, here you can see that there is a no hinge region. Okay. And but this has an additional immunoglobulin superfamily domain. In previous case, you have seen that there is only CH2 and CH3 is there. Okay. But in this case, you can see that there is a CH2, CH3 and CH4, and there is no hinge region. Okay. So, this is basically present in secreted immunoglobulin and membrane bound immunoglobulin, this kind of structure. Okay. So, immunoglobulin can be secreted. You know that immunoglobulin is secreted by plasma cell. Okay. Or it may be membrane bound. Membrane bound immunoglobulin is present in B cells. Okay. So, if it is a membrane bound, then the C terminal part of the constant region of heavy chain will have some additional thing that that here you can see that this C terminal end will have a extra cellular spacer which is consist of 26 amino acid. Okay. It is mainly hydrophobic in nature. So, most of amino acid, the 26 amino acid, they will be hydrophobic, and it is quite obvious because this is a transmembrane sequence. Okay. And there will be a cytoplasmic tail which will trigger the downstream signal, although this tail is very small. Okay. And this is not capable to activate the downstream signaling, that is why this is bound with some another molecule, okay, which we call it as a immunoglobulin alpha beta chain, okay, which is not which is required for downstream signaling. The bottom line is that. Okay. Here this is the structure of various immunoglobulin. Here you can see that there is a hinge region, okay, in case of IgG, IgD and IgA, okay. So this has a hinge region, okay, and here you can see the structure is quite different. All immunoglobulins are quite different, okay. Here you can see that in case of IgG there is a there are C gamma 2 and C gamma 3 region is there, and there is a predominant quite big hinge region in case of IgG, okay. And here you can see that IgA, they are present in dimeric form, and there is a some region is there which we call it as a J chain, which basically combines these two subunits, okay. And here you can see that IgM, it is present in pentameric form, okay, and this each subunit in this pentameric antibody is joined by J chain, okay. So now I will talk little more about the particular subtype. So IgM is basically present in pentameric form, okay. It is also present in monomeric form. IgM is also present in monomeric form and expressed on the B cells, okay. So the first antibody which is generated in against the antigen is IgM, okay, and on mature B cells IgM is present in monomeric form, okay. But when it is secreted, then it is secreted in pentameric form, okay. This is a first class produced in primary immune response or primary response. So that I have already told you. It can activate complements. I have explained you this has a more potential to activate the complement compared to the other antibodies, that is IgG, okay. This has a very good agglutination property, okay, this IgM. Agglutination is just something like it will cross link several molecules and make a kind of clump, okay, or agglutinin, okay. And this constitute about 5 to 10% of total serum immunoglobulin. Another is IgD. IgD is basically present in bound form on B cells, and this is the one antibody whose function is not very well understood, okay. This is present during the development. IgD is present during over the B cell during development or maturation of B cells, okay. We do not know what is the exact function of IgD, okay. Another is IgG. Here you can see that there is a very big hinge region, okay, and it is a most abundant immunoglobulin, and there are 4 subclasses of this IgG. If you remember the previous table where this molecular formula was given, over there you have seen that there is a gamma 1, gamma 2, gamma 3, gamma 4 heavy chains are there, okay. So therefore IgG is of 4 subtype, and you can call it as a IgG1 IgG2, IgG3, IgG4. Okay. These immunoglobulin can cross the placenta and give the protection to the fetus as well. They are involved in complement activation, but not all. I will show you in a short while which one is involved in activation of complement. Particularly IgG4 is not involved. I will show you in a short while. And it constitutes about 80% of total immunoglobulin in serum. Here this is the structure. Here you can see that the hinge region is quite variable, okay, in different subclasses, okay. So IgG3 has a quite huge hinge region, okay, and lot of disulfide linkages are there. This you can understand, okay. Another is IgA, which is in general present in dimeric form, okay. It is also present in trimeric or tetrameric form, okay. And this can cross-link large antigen, and this IgA is predominantly present in secretions, various secretion, okay, in our body, okay, and they give a protection over there. And daily production is quite high among other isotype. Every day about 5 gram to 15 gram is produced, this IgA, okay, in mucus, okay. So this is a structure of IgA. This has a secretory component or J chain, okay, and this is very important for the transportation. So IgA undergo transcytosis. This is transported across the membrane, okay. Here you can see this how the transportation of IgA is taking place. There is a polyimmunoglobulin receptor which is binding with this dimeric form of IgA, and then there will be a transcytosis, and then it is transported across the cell layer, okay. Last is IgE. So IgG is basically involved in various kind of allergic reaction, okay. It is involved in parasitic infection, okay. Basically this is increased during the parasite infection, and then at that time there is an increase in eosinophils also, and that condition we call it as a eosinophilia, okay. This eosinophilia is a kind of marker for allergy or parasite infection, okay. Now I will give you the kind of summary of all immunoglobulin. Here you can see that on top there is a molecular weight which you can see, and I need not to tell. Here you can see that there is a different kinds of heavy chain in different immunoglobulin. There is a gamma 1, gamma 2, 3, 4 is there, okay. Alpha 1, alpha 2 is there, mu is and epsilon and delta is there, okay. And there is a normal serum level is also given, and here you can see that IgG1 is present in quite high amount, okay. It's about 9 milligram per ml, okay. And in vivo serum half-life, so all immunoglobulin they have some half-life, and if you see very carefully the class of IgG, the subclasses of IgG, they have a highest half-life in the serum, okay. That is IgG1, IgG2 and IgG4. They are present about 23 days, okay. The half-life is about 23 days, okay. Which immunoglobulin can activate complement pathway? Here you can see that this IgG1, IgG2 and IgG3, they can activate complement, but not IgG4, okay. And the very good complement activator is IgM, okay. IgG1 and IgG2 can also cross the placenta, and they can give the protection to the fetus. Okay. In addition, IgG3 and 4 also can also move across the placenta. Okay. So, which immunoglobulin is present on mature cell? That is IgM. So, only IgM is present on mature cell. Okay. So, initially there will be IgM and which is present on mature B cell. IgM plus IgD both are present, and later on there will be one phenomena which we call it as a class switching, which I will discuss in subsequent session. Okay. So, this class switching changes the type of antibody. Okay. Some of these particularly IgG, they can bind with Fc receptor of phagocytes. Okay. And mucosal transportation is mainly take place for IgA. Here you can see that there are different both subtype of IgA can be transported in mucosal area. Okay. In addition, IgM is also can be also transported to some extent, and IgE is only which can activate the mast cell degranulation. Okay, you know that not only mast cell, eosinophil can be also degranulated. So, this is the overall summary of all different types of antibody, their structure as well as function. Okay. With this, I am stopping here. In next session, we will look at the antibody diversity, how the antibody diversity is generated, and another very important thing which we will learn is that how the one type of antibody is switches from one type to another type. So, we will discuss all those things. Although I will just touch upon all these topics, but I am not going to take you in a much more finer molecular detail. It's a huge topic. Okay, it will take a quite long time to finish. So, aim of this course is not that. Okay, thank you.